we already went over the role of the enzyme just as a catalyst in any particular reaction where substrate A is converted to a product. So this is the general reaction of enzyme catalysis, where the enzyme has to be specific, have a specific shape that it can bind that molecule, that substrate, and form a complex. Step. This enzyme substrate complex is not fixed. It can dissociate and reassociate, right? So the better connections via what kind of bonds? Covalent or non-covalent here? Non-covalent. So the better non-covalent interactions, the better the enzyme is going to be at binding that substrate, so the better fit of the shape of the protein for the enzyme, the better the complex, the more efficiently the complex will form. But that complex, no matter what, that complex will associate and dissociate, have a kind of constant rate of forward reaction and reverse. The timing then of the enzyme, so it has to be able to hold on to that substrate long enough for the product, for the actual chemical reaction, the bond, the change of energy, exchange of energy that occurs in converting the substrate to the product in order to facilitate or catalyze that reaction. And it does that by lowering the energy needed for A to be converted to B. So the enzymes are lowering the energy. That's now here, this is not A to C, the substrate is Y going to X. Okay? In an uncatalyzed manner, the energy necessary to be input may cause this reaction to occur at a very, very slow rate. And by lowering that activation energy, the rate is increased so that life can be supported. So how does this really happen?
So you can see the substrate in part A of this figure is blocked from being converted to its product by this barrier. But in the presence of the enzyme in the lower part, you can see that the barrier of the activation energy is reduced, and that's reduced by the presence of the enzyme holding the substrate in a way such that the activated intermediate is facilitated, and the reaction goes forward to produce a product. So you can think of this barrier like a wall that has to be overcome. It can be overcome by heat, but heat is not a useful form of energy. Instead, in the cells, enzymes help to reduce this barrier or this wall.
So I apologize, I'm having some trouble with the animations don't play. The QuickTime player is not working. So I'm going to invite you to
envision a molecule, here symbolized by the ball, that in principle can react in 4 different ways.
All right, here we go.
To undergo any of these reactions, the molecule must overcome an activation energy barrier that is of a characteristic height for each of the possible reactions. This can be achieved, for example, by putting more energy into the system, such as when heat is added. In the example shown here, the molecule will enter many different reaction paths by overcoming similar activation energy barriers. An enzyme, in contrast, reduces the activation energy barrier of only one specific reaction path. An enzyme, therefore, allows reactions to proceed at normal temperatures and directs them into one desired pathway.
Okay, so one way that we can speed up the reactions in a test tube is to do what? To heat it, right? So if you have a piece of paper that you light a match and it will burn very rapidly, and you'll be left with just the purely oxidized elements. But if you, in a cell, you don't want the reactions to proceed. Well, it can't tolerate the heat, right? So we know by homeostasis, by the rules of homeostasis, that cells have to maintain within limits certain conditions. And in addition, as part of those conditions, it's the temperature has to be maintained within a narrow range, right? So the cell is able to overcome the barrier by using the enzyme. And in addition to, you may have noticed that there were different pathways that the reaction could take in the case of the heated reaction. But really the enzyme is only catalyzing one of the possible ways that the activation energy can be reduced. And just back to my analogy with the piece of paper, really what the cell wants to do in converting energy and maintaining order within the cell is to cut up the piece of paper into many pieces that can then be utilized in other ways in the cell, rather than just burn it into a useless dust, right? So each enzyme in the cell you can think of as cutting up one piece of the puzzle so that, or the paper, so that there are useful products from each of those reactions. And all summed together, they add up to the metabolism of the cell. So how do enzymes find each other? So in your first chapter, there was an image along the top of one of the pages of just how crowded the molecules are and the organelles and compartments are inside a cell. And in that animation that we looked at, you could see there was a lot going on in the cell at any one time. So how do the enzymes find each other in a crowded cytoplasm? There's no directional movement to proteins. The only directional movement in a cell would be along the cytoskeleton, things such as transport vesicles that move molecules from place to place within the cell. But in the cytosol, okay, all those parts minus the organelles, there's just random motion. So a little bit of thermal motion. And so what ends up happening is that the enzyme and the substrate, just through their random motion, must meet. So what do you think are the limitation of the activity of an enzyme if they're just randomly floating through the cytosol of a cell until they their substrate and be able to hold on to it well enough? Juliana.
The number of bolts within the cell and their attraction to each other.
So there are
Strength
two,
of the bonds that make them.
right? So like we talked about, those non-covalent interactions that hold them together for any period of time, as well as they first have to come in contact with each other. So if there's not a lot of substrate around, the chances of that enzyme finding the substrate are pretty low. Because as we mentioned before, the enzyme, any one enzyme type of enzyme, glucose 6-phosphatase, there's only low levels in the cell, whereas we have lots of different enzymes within the cell. Remember, protein making up 15% of that 30% of non-water, the substrate, the molecules within the cell. Okay, so half of what's not water in the cell are proteins, and most of those are enzymes. But 15% out of the 30. Okay, so half of what's, yep, right? So remember there was, oops, this one's not going to work. So remember inside the cell you have 70% water. Okay, and 30% is other molecules, including the macromolecules, right? Okay. Yeah. Okay, and out of that 30%, okay, half of what that 30%, 15% of what's in the cell is proteins. So there are a lot of enzymes, but there's not a lot of any one particular enzyme because they're recycled. So great distances can be traveled through this random walking motion, Brownian motion, that the molecules have within the cytosol. So how do we actually measure how good an enzyme is, right? So if an enzyme is really good, even if the concentration is low, it's going to be able to catalyze that reaction. If the enzyme isn't that great, you're going to need greater numbers of substrate to get the reaction to go. And basically, how we measure the effectiveness of enzymes is by doing Michaelis-Menten kinetics, or enzyme kinetics more simply. Okay, so Michaelis was the biochemist that figured out that there's a constant that can, a measurable constant for the rate of reaction in the case of an enzyme reaction. So the Km, M being Michaelis, this is the constant that he figured out, is actually the concentration of, here on the x-axis, the substrate, okay, so what the enzyme binds. When the enzyme is at its half of its maximum speed, okay, so half of the rate of reaction. Okay? So let's look at a couple different possibilities. Okay, so for each enzyme, there'll be a different curve. Did you have a question, John? Okay. The curve will always be the same because the curve is basically measuring, so it'll have the same shape. because it's basically measuring the reaction when there's just very little substrate increasing over time, and measuring the rate. Okay, so intuitively, you know if there's more substrate, then that kind of random motion, going back a slide, that random motion, even if it only travels a short distance, if there's a lot of substrate around in the cytosol, the chance of it connecting with one of those substrate is much greater, right? But at a certain point, there's going to be a maximum that's reached. Okay, so these curves always start out looking like a line. Okay, very evenly dependent on the substrate concentration influences the reaction rate. At a certain point where the enzyme, all of the enzyme in that cell is bound with substrate, it doesn't matter if you throw in more substrate, nothing more is going to be converted because there are no more sites for the enzyme to bind. There's no more enzyme for the substrate to bind, right?
The enzyme concentration is not accounted for. This experiment was done for every enzyme, specific concentration of enzyme.
Specific, yeah. So if you change, that's true. The concentration is just kept constant. So at any specific concentration of enzyme 1 or enzyme 2, this is a kind of line or plot that you will find. Whereas first it's linear, and then later it will reach a maximum. Jesse?
So essentially, the higher up that it plateaus, the more effective it is. So the closer it is to when it's doing maximum rate of reaction, again plateauing, but if it goes up a little bit, then plateauing.
Right. So there are two things to look at. Okay, a very high Vmax, as Jesse just said, very high number of substrate that it can bind, very efficient. And also if it has a low Km. Okay, so you can see here has both a high Vmax and a low Km. Enzyme 1 is a better enzyme at doing its catalysis than enzyme 2, where it has a higher Km and a lower Vmax. Okay? So you can kind of think of this like if 100 people go to the grocery store and there's only one cart there, they're not going to be able to get their grocery shopping done very fast, right? They're going to have to wait for the next wait in line for the person ahead of them to use that cart, and then so whereas if there's 100 people but 100 carts, then that's fine. They can go at their usual rate and rapidly do their shopping. Okay, so this is how we measure how good or effective an enzyme is. And we can do this in the lab. We're going to do some similar reactions when we do the Hill reaction in lab. Any questions about that? So the Km is a constant that actually, so when you look at the enzyme substrate product, first reaction, the Km is used to describe kind of all of the constants of this reaction. The constant of the forward reaction, the constant of the reverse reaction, and the constant of the product being formed. Okay, so that's basically just the Km is describing all of these steps, because you can't individually measure these steps, but you can measure the Km when you have a fixed amount of enzyme. You're keeping that constant, you're changing, you have a kind of a titration of your substrate that you add to each reaction. And you actually just measure the product. In this case, you're measuring the light properties using a spectrometer, measuring the product that's formed. And you can calculate and plot the rate of reaction in that way. Okay, so as the substrate increases, the enzyme finds its substrate faster, so the rate increases. So there are two types of reactions, though, and enzymes can catalyze both in a cell. There are favorable or downhill reactions where the free energy, so G is the Gibbs free energy, delta G is the change in the energy, and delta G is going to indicate the positivity or the negativity of delta G. So basically the slope, downhill or uphill, is going to indicate whether the reaction is favorable. So if delta G is negative, it's a downhill reaction. The energy of the substrate is greater than the energy of the product. And so it naturally can move forward. The reaction moves forward, and the negative free energy indicates that disorder is increasing. So breakdown of molecules into smaller substrates are often downhill reactions. They're negative. So creating more disorder from organization or organized molecules, hydrolysis, etc. On the other hand, if the energy of the substrate is less than the energy of the product at the end of that reaction, then the delta G, or the change in the free energy, is positive. This is an unfavorable reaction. In order to get to that product, there needs to be some input of energy to help facilitate that reaction. But, so both happen in a cell, both favorable and unfavorable reactions happen in a cell. In the case of the unfavorable reactions, if you couple them to a favorable reaction, okay, so a lot of, one example that's given in your book is ATP as a delta G of negative 7.3 kcals per mole. Okay, so this is a very highly favorable negative delta G, right? Highly favorable reaction. And it's due to that high energy phosphate bond. the energy that's released from the loss of that phosphate. Okay? So by using that energy currency of the cell to couple it to unfavorable reactions in the cell, the biosynthesis of proteins, for example, any of those unfavorable reactions will be able to occur by coupling them. And that's because the energetically favorable reactions can occur spontaneously. And we have a lot of pathways that involve more than one chemical reaction. So think of glycolysis. With glucose, okay, first step to glucose 6-phosphate. Okay, but the end result of the catalysis of glucose is that you break it down to pyruvate. So this is just one of the many steps that go into this enzyme catalyzed pathway. And not every one of these reactions is energetically favorable. So some energy is required to catalyze the reaction. And by coupling the reactions, so if we just simplify it by saying A to B, B to C, okay? As soon as A is converted to B, it is ready to be a substrate for the next enzyme. Okay? If this is an energetically favored reaction, then it will drive the first reaction being unfavored, right? Okay, so if the delta G is positive here, or the delta G is negative. Okay, so that's one way that we can get unfavorable reactions to go in a cell. And one way that energy is or the order of the cell is energy, is maintained. So here in your book, they've shown just a cartoon that demonstrates the two types of energy, how kinetic energy can be transformed into heat alone, but if it's coupled to reaction or another event, that kinetic energy can be transformed to do work, and the water stored in the bucket then can be used to drive another reaction and produce useful work. So we don't want to just release heat from the cell. We want to do useful work in the cell, and that's really what coupling these favorable and unfavorable reactions together does. All right. Okay, so let's just touch upon chemical equilibrium for a moment. So we know that based on the Michaelis-Menten or enzyme kinetics, that substrate concentration has an influence on the rate of reaction. And so it's as the substrate is being converted to a product, there's going to be less substrate in the cell, right? So when you have a fixed amount of substrate and product, Okay? If the reaction is energetically favored, you will eventually, so if you have a lot of Y, then the product formation will be favored, and the arrow is drawn bigger for that reason. As the concentration of the substrate, Y, decreases, okay, then equilibrium will be reached and no net reaction. So the net movement here would be towards the product, but no net reaction would occur. If you look on it at the beginning, there would be equal number of X and Y. Fine, you have a favorable reaction. You're going to have a forward reaction. There will be a little bit at any one time of the reverse reaction, even if you start with those equal numbers. Okay, so over time, the ratio of the substrate and product will change. Then X molecules will increase and change that ratio until you get equilibrium. Okay? At equilibrium, it doesn't mean that the number of molecules are the same. It means that the rate of reaction is now equal. See, the arrows are now the same size. So it's equally likely that Y will be converted into its product as the reverse reaction. So the cell never reaches equilibrium at any one point inside the cell. This may happen in a test tube, but inside the cell, it never reaches equilibrium because of the coupling of reactions. X will then be used in a subsequent reaction. So what does the enzyme do to this rate at equilibrium? It basically speeds up, whether it's a forward reaction or reverse reaction. It's going to speed up the reaction, speed up the number of interactions between its substrate and hence the product. So in a metabolic pathway having multiple steps, X to Y to Z, here X is the substrate, Y is the product, and if it were reaching equilibrium, it would sling it to the next reaction where Y is the substrate for another enzyme. Okay? Even if there's a little bit of Y present, you're going to have the enzyme will increase the chances of both directions of the reaction occurring. But because that product won't build up, there'll be another step in the reaction. It will continuously go in the direction of, for example, glucose to pyruvate in the case of glycolysis.
So, in a pathway, like a metabolic pathway within the cell, while equilibrium will be reached between two molecules, the pathway is completed by linking or coupling reactions that are enzymatically catalyzed. So the enzyme that catalyzes X substrate to be converted to Y as a product, if linked to a reaction where the enzyme catalyzes Y to Z as a product, will help drive or siphon off the Y that might have accumulated if that equilibrium were allowed to be reached in the cell. So the enzymes themselves don't actually change the equilibrium point, or that ratio of 2 to 1 that you see here for X to Y at equilibrium. But it just speeds up the rate at which the reaction will reach that equilibrium point. By having a subsequent coupled reaction also speeding up that reaction, it pulls the product Y and causes the reaction to continue, or the metabolic pathway to continue to its endpoint.
In this way, each step will kind of pull the reaction in the proper direction, or in the intended direction of keeping order in the cell. So, besides the enzyme reducing the activation energy, we need to couple the reactions that are unfavorable to favorable reactions. And as I mentioned, one way is to couple a reaction with the dephosphorylation of ATP. But ATP isn't the only activated carrier molecule that can be coupled to unfavorable reactions to help them proceed. So you can think of catabolism, the breakdown, kind of naturally going towards disorder, as being energetically favorable reactions, whereas anabolism, or the building up of those macromolecules in the cell, are generally unfavorable. And that's why we're able to get energy produced in the form of ATP, the phosphorylation of ADP to ATP in the catabolic reactions, whereas the anabolic reactions require and use up that energy. They require that coupling to the energetically favorable reaction of ATP converted to ADP. So it's not only ATP that's an activated carrier molecule. You also have, so in this case, ATP is carrying a very high energy phosphate. But you can also have molecules like NAD+, FAD+. Okay, these can be reduced to NADH form, are electron carriers, so they carry a lot of energy by carrying energetically active electrons that can be easily given up to do useful work in the cell. So in the case of ATP, we already talked about how ATP can be generated either by photophosphorylation from the sunlight or from food, so by substrate or oxidative phosphorylation in those cells that don't carry out photosynthesis. And it's really these two phosphoanhydride bonds between the adenosine monophosphate and diphosphate, as well as the bond between the ADP and the ATP, diphosphate and triphosphate. These are both energetically favorable reactions, but in the cell, what usually happens is that the ATP loses one phosphate group and releases enough energy to make the energetically unfavorable reaction favorable. Okay. So really, when we write a reaction that requires ATP, it's really, we haven't written out the reaction of ATP converted to ADP, but that's really the second reaction that's being coupled. Okay, so in the case of a condensation reaction, or building those macromolecules, building the proteins from amino acids by making that peptide bond. For example, are in general energetically unfavorable, and the hydrolysis or the breaking of those macromolecules into their subunits by the addition of water are energetically favorable. And ATP is what's used to facilitate the energy from ATP is converted to help those unfavorable reactions become favorable as far as their free energy. Okay, so here's an example of ATP used in the condensation of or dehydrogenation of a polysaccharide to in a monosaccharide or disaccharide to a polysaccharide. So in the growing chain of glycogen, when a monosaccharide is added, condensation reaction using the energy from the release of that last. Okay, so it doesn't show that second reaction here. It's only showing the unfavorable reaction here. But together, it will enable the building of the macromolecule, the polymer of glycogen. And I already mentioned the example of proteins, which also would similarly, from the ribosome as it adds, as the tRNA is bringing in that additional amino acid, it's going to catalyze, it's going to bring the growing peptide chain in close proximity to the amino acid so that the condensation reaction can occur to create the peptide bond. So a new peptide bond in the growing chain of the polypeptide. So when we look at the two reactions separately, it's actually happening in two steps. The missing reaction in those previous figures that I showed is really the ATP transferring a phosphate to the intermediate, whether it's to polysaccharide or whether it's an amino acid. So B represents that subunit. Because phosphate group is unstable, it's then lost, and the bonding, the chemical bond, the covalent bond between A and B is created. Okay, so the P here represents the phosphate group, the PO3 group that would be lost from ATP and released as a product of the condensation reaction. So here it would be the inorganic phosphate. All right, so I mentioned ATP is not the only ACM or activated carrier molecule that can help unfavorable reactions proceed. So in the interest of time, I'm not going to go over the actual covalent bond that occurs as the intermediate. So this is just from your book. It's the next form, the BOP of the subunit being phosphorylated, that intermediate form. That would then be unstable and form the product, the covalent bond in the condensation reaction. Instead, I'm going to go on to just mention a couple other activated carrier molecules. Okay, so one result of catabolism is ATP. They're really important electron carriers. They result from the glycolysis, the process of glycolysis, for example. And NADPH is one of those. Okay, so NADPH is that, thinking back to last time when we talked about oxidation and reduction, is NADPH oxidized or reduced? It's going to be the reduced form. So it's the one, so remember the reduced is gaining electrons. So that is the high energy. It happens that these electrons that it gains are high energy electrons, and that NADPH is ready to give up its electrons to facilitate unfavorable reactions. Okay, so whether it's NAD, which just is the same molecule without the phosphate group, or whether it's NADPH, the electrons that it carries, okay, and you can tell that along with the electrons, the hydrogen is gained. So you can tell that the reduced form is the form with the hydrogen. Same thing for NADH. NADH, NADPH, that's the reduced form. Okay, and this is what the chemical structure looks like, but it's really that unstable bond here, those electrons that are reactive and can be readily given back to then convert NADPH or NADH back to its oxidized form. So you can think of NADPH as reducing, it has reducing power in that it has the ability to reduce other molecules by giving up those electrons. Okay. So one example would be the reaction of 7-dehydrocholesterol. Okay, so in the lipid biosynthesis, NADPH catalyzes, helps to make this reaction energetically favorable. Okay, so here we have a carbon-carbon double bond. So double bonds being a single bond. Those double bonds these are really strong bonds that are unfavorable to break. But by giving up those electrons, the double bond is broken to a single carbon-carbon bond. Okay, so I just have about a minute more of your time So I'm just wondering if there are any questions from that. You should be able to explain in general those coupled reactions with the example of ATP. And I have some practice problems that I've listed here that are in your book. Again, the practice problems are, the answers are in the back of your book. And also there's a practice that you can find as the Chapter 3 quiz, so those multiple choice questions. Just a note about the exam, because it is coming up a week from today. The format will be some multiple choice, but mostly a short answer. And you should bring a calculator, because I will be asking you to do some calculations similar to what we already did in